So question 17 in the chapter 16 homework is talking about a protein named trypsin. So trypsin, and just so we can appreciate the beauty of trypsin, let's go to the protein data bank. Here's what trypsin looks like in 3 dimensions. Those gray spheres are carbons, the red ones are oxygens, blue ones are nitrogens. So that is trypsin. That is a peptidase, and that means it's an enzyme because it ends in ASE that chews up peptides. And specifically, it hydrolyzes. Remember, lyse means to break apart using water, hydro. So it's going to break apart polypeptides. Those are proteins. You have a lot of peptides brought together. So this is going to sort of act like little scissors that cut up a long protein. That's what trypsin does. And it does that in your small intestines, which makes sense, right? You eat proteins from plant sources or animal sources. They make it to your small intestine. Ultimately, you have to get them down to single molecules before your cells can break them down. Enzymes like trypsin break them up. Now, what they're saying in this problem is that trypsin works at an optimal pH. So it works best at a pH of 7.7 to 8. In other words, the enzyme works best at that pH range. Now, enzymes speed things up. So the reaction will happen fastest in the pH between 7.7 and 8, because that's when trypsin will be working best, and what trypsin does is to speed up this reaction. So the reaction will happen fastest in that range. Then it says, how is the rate of a trypsin-catalyzed reaction, so a reaction that's sped up by trypsin, how is the rate affected by each of the following? And it says changing the pH to 3. 3 is way out of the range. That's the best. And so what'll happen is that solution, which is very acidic, will squeeze the trypsin molecule into a shape so that the active site doesn't correspond as well to the substrate. So trypsin unfolds and can't speed up the reaction anymore. Trypsin unfolds and can't speed up the reaction. Won't fit into the mouth of the Pac-Man. The active site, if trypsin unfolds, then the active site doesn't have the same shape, and the substrate can't fit in there. And so the reaction happens more slowly. The rate of the reaction slows down. What would happen if you ran the reaction at 75 degrees Celsius? Now, the temperature in your small intestine is around 35 degrees Celsius, which is around 97 degrees Fahrenheit. So imagine what that feels like to be out on a day that's 97 degrees Fahrenheit. That's the temperature inside your small intestine. This is 75 degrees Celsius, which is around 167 degrees Fahrenheit. So at that point, just like we would start to melt, and the same sort of thing happens to this trypsin. At that high of a temperature, all of the molecules surrounding the trypsin, the water molecules in solution, knock against it really, really hard and really, really fast, over and over and over again. And so that bombardment of the trypsin by all the molecules around it at this high temperature makes trypsin unfold again. And so yet again, trypsin unfolds and can't speed up the reaction because the substrate won't fit into the active site anymore. And so the rate of the reaction slows down. Now, if they had given any pH outside of this range, it doesn't have to be it would slow down the reaction because the same thing would happen. Trypsin would start to unfold, and so it wouldn't be able to eat up its little Pac-Man food as fast. The substrate molecules wouldn't fit into the active site as well, and so the reaction would slow down. And likewise, if you made a really, really low temperature, then trypsin, the reaction might also happen more slowly because trypsin is crafted to function at the temperature of the small intestine, which is around 36 degrees Fahrenheit, or 36 degrees Celsius, which is 97 degrees Fahrenheit.